Hi, Aravind. Welcome to the third part of this series on the discovery of apoptotic domains and their roles and their evolutionary histories. In the second part, you had laid the foundation uh, for the of the early discoveries of these domains that are involved in apoptosis and what it actually meant to see them in prokaryotes, and what was the thinking at that time about their role and their diversity in eukaryotic systems, and their role in prokaryotic systems. Also, after that, I know that there were a lot of follow-up experiments based on some of those early discoveries and identifications of domains. But science has never been linear, and in order for us to have come all the way to have a much better understanding of what we know today about apoptotic-like systems in prokaryotes, we had to undergo a journey which involved many other diversions, which all contributed to the final big picture. So I think if you can sort of illuminate us on those particular aspects through which we had to go in order to reach where we are today, that would be great.
Yeah, so by around 2003, which is roughly the time frame which we have covered so far, uh, we pretty much had exhausted to the extent we could with the sequence availability at that time, that is the state of the database at that time, and the tools for sequence analysis which we had. We had pretty much reached the end point of understanding from a domain-centric viewpoint uh, metazoan apoptosis. So the basic domains had been discovered, the domain architectures had been discovered, at least in those metazoans which we had till that point. And some broad mechanisms which could be predicted based on sequence analysis had also become clear, which included, to just recap, the homotypic interactions between domains of the death-like superfamily, the peptidolytic effector systems based on caspases, and the regulatory systems based on the stand class of NTPases. And this pair of the anti-apoptotic and pro-apoptotic components working antagonistically to maintain a certain balance, which came from the BCL2 superfamily. Mm-hmm. And the origins of most of these had been sort of tied to the prokaryotic world. We had very clear prokaryotic homologs of most of these domains, barring two domains, which sort of set the stage for the future analysis: the death-like superfamily and the TRAD-N family. We'll come to these down the line. So these could not be traced back to the prokaryotes, but the catalytic domains, in most part, could be traced back to the prokaryotes. And it was clear that they were coming together in prokaryotes in some way, but did they constitute an apoptotic system in prokaryotes? That remained completely uncertain at that point. And there was also a lot of murkiness about the nature of prokaryotic apoptosis itself around that period. So one could say that at that point, I took a break from focusing on apoptosis proper. From my perspective, what could be done was mostly done. And maybe there were some low-hanging fruits which could have been picked, but I just left them alone and others picked them up. But one thing which happened was wet lab confirmation of some of the hypotheses. For example, the peptidase activity of the paracaspase was confirmed in the first decade of the 2000s. And subsequently, it was established that the metacaspases indeed play a role in apoptosis in plants. So our general understanding regarding those in terms of our predictions and their wet lab confirmations sort of extended the basic understanding. But at this phase, I moved back to some other questions which had long interested me. As you know, we have always discussed the proteomes of organisms as having two uh, evolutionary trajectories. There is one part which we call the core. The core of the core, so to say, is the translation apparatus, which is highly conserved, and you can see the signal of the last universal common ancestor from which all organisms alive today on Earth have descended in the translation system. Then, somewhat uh, similarly conserved is the core of the transcription apparatus, the RNA polymerase, which you were very much involved in the studies which we did on that. And then there were the DNA replication enzymes, which too you were very much involved in studying. They are even less conserved than the transcription apparatus, but still you can consider them as forming part of the core because they generally show a high degree of vertical inheritance. And you can discern the phylogeny of organisms in this core. But the majority of the proteome is not this core. If one were to go by the core, you would be left with just a small number of proteins as strictly traceable to the LUCA. But definitely LUCA was not a stripped down organism with just a few genes. We have strong reasons to suspect it had a so-called periphery, like all organisms today have, wherein you find the majority of proteins which are much less conserved. And a good part of this periphery comprises of things which may be seen as metabolic in nature, because the metabolism of organisms is selected by their very specific environmental niches. And depending on those niches, you have a particular enzymatic repertoire or a sensory repertoire to sense external stimuli in the form of small molecules and photons and the like. So a part of the time was spent dissecting those sensory and metabolic aspects of this periphery. But there is one part of the periphery which interested me considerably, and I felt there was a lot to be learned there, which is that part of the periphery which is devoted to biological conflicts. And here is where our interest in this concept of biological conflicts began, which very simply can be described as a conflict between two different entities, genetic entities. And it can occur at all levels. Like the first thing which we focused on was the conflict which occurs within genomes. So you have entities like these toxin-antitoxin systems, which till that point had been studied and seemed to be a very disparate group of systems in bacteria, which have two genes typically, one coding for a toxin, which is generally stable, and another coding for an antitoxin, which neutralizes the toxin and typically tends to be labile. And thus they keep, the antitoxin keeps the toxin in check. But if the gene pair were to be broken and the one of them lost, since the antitoxin is labile and is degraded quickly, the toxin, if it's lost, it would still, the protein will still persist and kill the cell. So there's a tendency to maintain that pair. And if the pair existed on a plasmid, there is addiction of the plasmid, which we discussed in the first part. But this is a conflict between these elements in the genome. But at the same time, one could imagine that they confer a certain advantage to the cell through the apoptosis process. So in a sense, they have this two-faced nature, where they are selfish elements at the basic level, but they sweeten the pie for the host genome to a degree by conferring some advantages. And this where the equilibrium is would depend on a variety of selective forces acting on the organism. So there may be certain selective forces which tip the balance towards the so-called domestication of these toxin-antitoxin systems, where they can become more regulatory in nature as opposed to being purely selfish. And there can be other scenarios where they can proliferate in the genome quite like a transposon, which is even more of of an outright selfish element, and colonize the genome in multiple copies where they are exhibiting their selfish tendencies to a greater extent. But the truth seems to be some kind of a balance between the selfish side and the advantages which they confer. So one of the programs which I initiated was to use computational methods to discover new toxin-antitoxin systems. And the basic idea here was to discover some kind of a grammar which these toxin-antitoxin systems had. And the grammar in this case was relatively simple because it was just two genes. And there tended to be a common but not universal tendency of what were called the so-called type 2 toxin-antitoxin systems, where the antitoxin tended to be a transcription factor, which in addition to binding the toxin could also bind DNA and regulate transcription. And the toxin tended to be an effector, most often of the catalytic type, but they could also be other kinds of non-catalytic means of cell killing. So by a combination of sequence analysis, where we could identify a catalytic effector combined with this non-catalytic component, which could serve as an antitoxin, the genomic syntax could be established. And you could search for gene pairs which fit this pre-established syntax. And using that method, for the first time, we discovered many new toxin-antitoxin systems. And that method became a favorite for people to repeat and redo over the years several times to find new TA systems. But in this first work on TA systems, where we first presented this method and discovered new systems, one new toxin which we found among several others was the PIN domain. And this PIN domain is a metal-dependent endoRNase, which cuts RNAs essentially. And this, we had earlier noticed this PIN domain in eukaryotic systems, such as the nonsense-mediated decay system, which is involved in degrading RNAs containing nonsense codons. So here suddenly we had an evolutionary connection between two seemingly disparate systems. One, the selfish toxin-antitoxin system, and the other, this eukaryotic housekeeping system in a sense, which clears these defective mRNAs with nonsense codons. So that pointed to an evolutionary connection for the potential origins of this NMD system in the eukaryotes through the domestication of one of these ancient toxin-antitoxin systems. In the same study, we also extended them to the other great branch of the prokaryotic morphology, that is the archaea, and showed that they were very prevalent there and might combine with some other components, giving the first hints that they may be involved in some kind of a domesticated immunity related process. The next notable study which we conducted was that of the prokaryotic ubiquitin systems. So this again stemmed from this question of the periphery and the core. Now, in eukaryotes, one of the core systems which we saw at that point was the ubiquitin system. And we had discovered some very important components of it, such as the proteasomal deubiquitinase, the JAB domain. So right at the time I discovered the JAB domain, I realized that there were prokaryotic homologs of it. And but there was no obvious prokaryotic ubiquitin system. So that sparked this project to find the roots of the eukaryotic ubiquitin system. And it was a project which lasted several years, and our understanding of it unfolded in a series of steps. But one of the things, again, which relates to this toxin-antitoxin work is the establishment of certain syntaxes. So we could show that these prokaryotic ubiquitin systems show some syntaxes which were suggestive of a diversity of ubiquitinating and deubiquitinating systems. In their very simplest form, they just had the first enzyme, the ubiquitin conjugating E1, and were accompanied by a JAB domain in some cases. And these seem to be involved primarily in systems which were more of a metabolic type, in secondary metabolism or in the biosynthesis of cysteine. Right. And then there were slightly more elaborate versions, which we found in the first study, which had the first prokaryotic versions of the E2 enzyme. And here we could clearly predict protein ubiquitination was underway. And it took us some time before we finally discovered the prokaryotic ring fingers, the E3s, which suggested that the whole triligase system, along with the deubiquitinating JAB domain, was present. And these were present in bacteria which did not have the conventional proteasome, and nor was there a linkage necessarily to the proteasome in bacteria and archaea which had such systems. So it suggested that this prokaryotic ubiquitin system emerged completely independently of the proteasomal system, and they came together only in certain lineages and got fixed in the eukaryotic stem lineage as the proteasome ubiquitin system.
Right. And previous to, previous to the discoveries of E2 and E3, they were known to be, or they were shown to be involved in synthesis of those cofactors, thiamine and molybdenum.
Yes.
So that's what they thought the origins was like. This is where it originated, and then in eukaryotes you had this assembly and evolution that took place that led to the ubiquitin system. But I think
right, where URM1, an ortholog of the TS MOAD group of the prokaryotic world, is conjugated like ubiquitin.
Right.
So it was clear that there was a connection, but that there were complete systems, a triligase system and a JAB, working independently of proteasomes in prokaryotes, conventional eukaryote type proteasomes to be very specific, was one of the key things which came out of that work of ours. And it suggested that these two had distinct pathways. There was the proteasomal system for protein degradation, and there was the tagging system through ubiquitin, which emerged separately. Now, there were two lessons, one which took shape only later, and the other which immediately came to mind when we studied these, when we discovered these UB systems in prokaryotes and studied them. So the key lesson was that they were very mobile, and it suggested that they were fast evolving, and in many ways, this clustering of the ligase, the deubiquitinase, and some other genes linked to them which resemble restriction endonucleases or pin-like RNases, suggested that there may be a role for these in biological conflicts, and that these ubiquitin systems, right then we predicted, were some kind of analog to the restriction modification or CRISPR-Cas systems in being mobile entities involved in some form of biological conflict. The second lesson, which took us a while to unravel, was that these ubiquitin systems may be connected to certain enzymes which are generating nucleotides, signaling nucleotides, which are related to the DNA polymerase beta superfamily, which is a large superfamily of nucleotidyltransferases, which were first discovered by Lisa Holm in the 90s. And subsequently, I studied them quite a bit and discovered many new families of them, and established this link between polymerases and nucleotide cyclases, or 25A oligonucleotide, signaling nucleotide generating enzymes. So in that UB work, we realized that there was some connection between these systems, which took us some time to unravel, but it was sort of like the stepping stone for that future work. The third major development which took place was our discovery of the polymorphic toxin systems, which we approached entirely through a computational angle and discovered it more or less simultaneously as their first wet lab report, which found them to be associated with this phenomenon of contact-dependent killing, where within bacteria, two bacteria may kill each other upon contact by using cell surface toxin. And these toxins show polymorphism in that there is a recombinational process which causes the toxin to change within the toxin locus through recombination with cassettes, which have standalone versions of other toxin domains. So our systematic analysis of the system or related systems revealed several important things. In a sense, it was to lay the groundwork for the future understanding or the more complete understanding of the origins of apoptosis systems. So to quickly summarize the key discoveries, we found that now we could extend this concept of the toxin-antitoxin like pair to other systems which involved toxin effectors, but they were not intracellular, but they were cell surface. So they were part of a different level of conflict. It was not conflict within the genome, but conflict between organisms. In this case, the classic polymorphic toxin systems are involved in conflicts between organisms of the same species. So you can consider them two distinct strains, if you would like to use that term, each of which is encoding its own toxin, and they exclude each other. So here we add a determinant which could finally explain the phenomenon of kin recognition in bacteria and some archaea which had related systems. And this kin recognition process could hence lay the groundwork for understanding the importance of kin selection in bacteria. So when you're producing shared goods like siderophores and secreted enzymes, you only want to share them with kin, so you want to exclude non-kin. And these polymorphic toxin systems provided an excellent explanation for how that conflict would play out, with the exclusion happening through these cell surface or secreted toxins. And the way they prevent self-intoxication is through an immunity protein. And it was these immunity proteins which greatly interested Dapeng at that point, which got us into studying these systems. And uh, both the toxins and the immunity proteins had an evolutionary consequence way beyond these systems. So just to outline a few of those, we discovered that these immunity proteins have contributed to, say, the hedgehog signaling pathway of metazoans. So there is this protein suppressor fused, which is part of the hedgehog signaling pathway, which also we had been studying to a degree in the past and subsequently with the wet lab collaborators. But that's just an aside. But the origin of some of those components were a mystery. And here we could show that SUFU, suppressor fused, had emerged directly from one of the immunity proteins in this polymorphic toxin system. Similarly, the polymorphic toxin tips of and why I call them tips are because the toxin proteins tend to be large proteins, which have N-terminal domains associated with secretion, central domains associated with encasement, display, and proteolytic processing of the toxin. And finally, the C-terminal tip of the protein, like the tip of a spear, is what contained the toxin, which typically tended to be catalytic. And so some of the toxins which we discovered there included the nucleic acid deaminases. So that gave us the answer for the origin of something which was a long-standing mystery for us. That is the origin of the AID-ApoBec-like deaminases, which are antiretroviral restriction enzymes in mammals, for example. And they are also involved in the mutagenic diversification of the immunoglobulins in vertebrates, in the jawed vertebrates. And a related version of it is involved in the mutagenic diversification of the VLRs in the jawless vertebrates like the lamprey and the hagfish. So these were things which we had studied earlier, and so we had encountered AID and ApoBec repeatedly, but where it came from was a big mystery. And in one stroke, we were able to solve that great mystery with the discovery of the polymorphic toxin systems, because one of the toxin tips was this AID-ApoBec-like deaminase, and we could show that there was a great radiation of these deaminases in the polymorphic toxins, from which the AID-ApoBec-like deaminases had emerged. And in fact, we even found a smoking gun in the form of one of the Wolbachias, which possesses a similar kind of toxin. In this case, it's not involved in interbacterial conflict, but deployed against its host in all likelihood, which contained that AID-ApoBec-like deaminase. So it was through such endosymbiotic bacteria, it was likely that the AID-ApoBec-like deaminases were acquired by eukaryotes on more than one occasion. One in the metazoan lineage, another in the dictyosteliid slime molds, and there are yet other examples amidst the general SAR lineage of eukaryotes. So this was one case. And the other notable thing was the discovery of several H&H domain endonuclease toxins in these polymorphic toxin systems. And the H&H domain, just to give some background, is an metal-dependent endonuclease domain, which contains a version of the treble clef fold. So those H&H domain toxins, we could show when now the progenitors of an important apoptotic enzyme, that is the apoptotic endonuclease of various animals, metazoans that is, which is involved in cleaving the DNA endonucleolytically, which is one of the final steps in the apoptotic process, the destruction of the nuclear DNA. by this endonuclease. And now we could establish that its roots also lay in this polymorphic toxin system. As we discussed the last time, we could trace the zoophy domain back to these polymorphic toxins, where it plays the role of a prokaryotic processing domain. And we could also trace the origins of these ADP ribosyl transferases, ARTs, of the carp and pyrazine-like families, which play a role similar to this apoptotic endonuclease, the CAD endonuclease, in certain insects and mollusks, like in some uh, lepidopterans, in certain moths. You have this ART as the enzyme which works in apoptotic targeting of the nuclear DNA through ADP ribosylation. And likewise, in those molluscan systems, CARP plays a similar role in ADP ribosylating DNA. And what we showed was that there were two distinct origins for CARP and ADP and pyrazine, these two ADP ribosyl transferases, from two distinct types of polymorphic toxin tips. And just to linger on this a little more, maybe one more notable example of such a player, we reported several cases of eukaryotic seeding, so to say, is this SGS domain, which is a toxin which we now suspect could be an endonuclease, which was transferred from these bacteria to eukaryotes, and it's the salivary gland toxin of mosquitoes. And now there's a very recent preprint which suggests it may play a role in the facultative sex ratio determination in Daphnia. So that's another example of one of these toxins from the bacterial world, from the polymorphic toxin world, crossing over to eukaryotes and being reused in certain processes.
Yeah, and the famous one, the tenurin, I remember.
Yeah, yeah, tenurin is interesting in that it has been, the toxin domain became inactive there, and it was used as a signaling molecule in neural axon and dendrite contact processes. So that's a shift. You can say it's an exaptation rather than a direct use of the toxin activity.
Yeah.
There's one more example which we could cite, which is more of a direct use, which is the ADP ribosyl cyclases, which again is an activity comparable to the ARTs, which use NAD to create the cyclic ADP ribose. Just like the ARTs use NAD plus as a substrate to transfer the ADP ribose to a substrate, like a protein or a nucleic acid. In this case, the same NAD plus is used to just make a cyclic derivative of it, or NAADP, which it can be made from NADP, another kind of derivative. So those ARC enzymes, which are part of, which are found in metazoans, and they're part of the immune system of vertebrates, we could clearly establish their origins again in these polymorphic toxin systems. So that's one more of the toxin tips. So to summarize, we could show that a variety of toxin domains had been reused by eukaryotes, especially metazoans, in capacities where the toxin activity was used for something distinct from the prokaryotic conflict, where it was a conflict between two bacteria of the same species, or a conflict between the bacterium and its eukaryotic host, or between two unrelated bacteria, an interbacterial conflict. So that it was not used in that kind of a context, but it was reused often in an intracellular conflict uh, context to target invasive elements like retroviruses in the case of the Apoptotic clade of deaminases, or in an apoptotic process to kill the cell, probably when under attack from an invasive uh, entity. So this filled up some important gaps which we had failed to figure out when we last dropped off our directed studies on animal apoptosis. That is, where do these effectors, which actually target the DNA and bring an end to the cell, those very final steps, where did they come from? So finally, we could establish a bacterial origin for them, but it was very different from the bacterial origins which we established for, say, the stand or some of the other domains, um, which did not seem to be in any particular toxin systems. In fact, what they were doing in prokaryotes was quite unclear. And those prokaryotic homologs, which we had some functional idea of, did not seem to be anything related to apoptosis. So one more brick was added, you may say, in building the edifice. But there was another important lesson which this study of the polymorphic toxins taught us. And that was when we did a systematic comparison with the toxin-antitoxin systems. And in the middle, we had also engaged in some studies on other conflict systems like secondary metabolism systems uh, involved in the synthesis of antibiotics and siderophores and the like. And so what that revealed was that there were a series of enzymes in those systems, which again had interesting crossovers to the eukaryotic side. Like we showed quite comprehensively that these were the founders of many eukaryotic protein modifying enzymes in chromatin, such as the SET domain, which is the histone methyltransferase, and some other histone methyltransferases like DOT1 and PRMTs, the arginine methyltransferases, that their ultimate roots lay in these secondary metabolism systems, which are also deployed in interbacterial conflicts through the production of toxic compounds like antibiotics. So, synthesizing all these conflict systems, we found that there was one remarkable thing. Even though they worked at very different levels or through deployment of very different kinds of molecules, there was a sharing of what could be termed the effector components. So we found some of the same endoRNases, like the RELE like endoRNases, which we unified along with Barnase and others into this Becker superfamily. They were big players in the toxin-antitoxin systems. They were also the primary endoRNases in the polymorphic toxin systems. And the same could be said of the restriction in endodnases, the REases. The restriction endodnases were the primary effectors in the RM systems and the CRISPR-Cas systems to a degree, some of them at least. And here again, in the polymorphic toxin systems, one of the major effectors were these endodnases or the REase superfamily. So what it told us was that there was a lively exchange of these effectors between different conflict systems. And even though they may be deployed in different contexts, the ultimate objective of cell killing was achieved through a similar mechanism. And what that mechanism was targeting the central dogma. So the systems of the central dogma, the core which we discussed earlier, is what cells have difficulty evolving away from. That is, there is a lot of functional constrained. So much of the natural selection acting on it is of the purifying type, and it tends to conserve sequences. So the ribosomes, ribosomal proteins and RNAs, uh, topoisomerases involved in replication and repair, or RNA polymerases, they tend to show a high degree of conservation. And so you have a tendency to target the nucleic acids through, or the nucleic acid binding proteins, like the ribosomal proteins, through this central dogma system. And that's where you have the sharing of effectors, or that was one of the major driving forces in the sharing of effectors between these diverse conflict systems. And that from each of these systems, the eukaryotes had repeatedly acquired um, some of these systems, some of these domains, and reused them in a very innovative fashion, often not in the same way as they were used in bacteria, but in immunity or in modifying uh, proteins in chromatin for epigenetic modifications. So, as a result of these studies, we had gathered a large body of grammar, one may say, of these systems. Uh, and I cannot but avoid mentioning the analogy to our tradition, where the great grammarian Parnini is said to have journeyed all over the Sanskrit speaking world to have gathered usages to reconstruct the common grammar, which then became the norm for the language.
Right.
So, in a very similar way, we had journeyed across the space of conflict systems and had gathered all the usages, and we could now distill it down to a degree of uh, uh, interpretable grammar, and now use that to discover new systems. And the first major foray in this direction led us to what may be called the nucleotide dependent conflict systems. And this was the fourth major development in our group. And it involved the discovery of systems which used either nucleotides or their derivatives. And these can span a very wide range of molecules. For example, they could be just the base, which is clipped off from the nucleotide, or a completely novel nucleotide, which is synthesized by the tear adjacent biosynthetic systems, or the more common ones which were seen in eukaryotic metazoan systems till that point, but not outside of them, um, like the cyclic dinucleotides, metazoan conflict systems. They had been seen in prokaryotic signaling systems where they were recognized by a ribozyme, like those generated by the GGDF domain. But in the eukaryotic immunity, the 2,5-A oligoadenylate was a major signaling component, and so also some cyclic dinucleotides. So this work allowed us to extend these nucleotide-based signaling systems back to the prokaryotic world. And in the prokaryotic world, we found that there was a great diversity of these, and those were again the seeds of the metazoan immune systems which used these nucleotide signals. For example, we found the progenitors of the oligoadenylate, the 2,5-A oligoadenylate systems in bacterial conflict systems, and then we found progenitors of the various cyclic dinucleotide systems again in the prokaryotes. What was notable in these systems was the emergence of this principle, which we term the thresholding principle. So, why do you involve a nucleotide which is synthesized at great cost, that is, consumption of ATP or GTP in order to activate the effector. So very quickly to caricature these had three components: a synthetase, the synthetic or the generating enzyme, which would generate this nucleotide or nucleotide derivative, and then a recognition domain or a sensor domain, which would sense that nucleotide product, and finally the effector domain, which was often catalytic, but in some cases potentially pore forming, as they were membrane proteins, which would then affect a biological conflict action. So this tripartite organization meant something uh, very significant in terms of how they worked. First, let's deal with the effectors. What we found was that these effectors were closely related in most part to the effectors which came from other conflict systems. So we had already had a sampling of effectors in these toxin-antitoxin systems, restriction modification systems, CRISPR-Cas systems, and the polymorphic toxin systems. So here again, the effectors are in a similar vein. And to name a few, the restriction endonucleases, caspases, the Becker-like RNases, and the TIR domain, which we will dwell upon in some detail, which we had earlier encountered in the apoptotic systems too. Right. So all these were the effectors. But what was unique to these was that synthetase, which generated nucleotide, oligonucleotide, or a cyclic nucleotide product, and in some cases, a derivative of a nucleotide. More generally, these could be called generating enzymes because we could predict that some of these were using NAD plus to generate an NAD derived signal rather than using a conventional nucleotide triphosphate that's like ATP or GTP. And then there was the sensor domain, which would sense that signal, which is coming out from these the signal generated by the generating enzyme. So what we could say is that these came under a common umbrella, wherein the generating enzyme created an intermediate signal, the second messenger, so to say. And the sensing of that second messenger was the one which unleashed the effector. So this suggested that there was a threshold which had to be surpassed in order for the effector to be deployed. And when we look at it from the viewpoint of apoptosis, it suddenly started to make sense because most of these effectors are likely to have a deleterious effect on the cell, even if some of them were to be directed at the invasive nucleic acid. Because the discrimination of self versus non-self nucleic acid is not always an easy thing. And there was no evidence that these were working under precise self non-self discrimination like the prokaryotic RM systems. So there was always a deleterious aspect to the cell in effector deployment. So if you had these systems firing off at the slightest trigger, then you're likely to face a downside to it, or it would be disadvantageous because you would be losing cells and perhaps triggering an inflammatory process if we were to look at a multicellular organism like a metazoa. Whereas in a prokaryotic system, it would be just a total loss of fitness with the death of a single cell if this system was triggered at the slightest of the impulses. That is the input signal was very slight and you triggered the effector. So by placing a nucleotide in between, you had to cross a certain threshold, and it was only when a nucleotide was generated would this signal be reached. It would imply that a sufficient threshold had been reached for the effector to be deployed. So that way, one could ensure that the death of the cell was not did not take place in a frivolous manner with the slightest of signals of an invasive entity. And the other thing which came out, an evolutionary observation, was that repeatedly we observed that these signal generating enzymes had connection to enzymes which may be involved in RNA repair, like the Pol beta superfamily nucleotidyltransferases, or in recognition of DNA during conjugation, like the slog superfamily. And in some other cases, in the utilization of NAD, like members of, we were able to unify slog with TIR and another family, the deoxyribonucleotide hydrolases, the DR hide for short, superfamily. So the unification of all these as a clade of rosmanoid domains which bound nucleic acids or nucleotides, including NAD, brought them together. So the signals, they had a link on one side to nucleic acid biology, that is nucleic acid repair, sensing of nucleic acid during conjugation or transformation, as in the case of the slog superfamily. And on the other side, as these signaling enzymes in these conflict systems. So that suggested that the existence of nucleic acid damage or the sensing of the entry of nucleic acid from outside was a phenomenon which probably served as the foundation for the emergence of these enzymes. So they probably created some byproducts which then acted as signals for deployment of effectors in the conflict with the invasive entity. And these got institutionalized as enzymes which now generated a dedicated signal upon detection of the invasive entity. So putting these together, we could come up with an evolutionary model for how these nucleotide dependent threshold setting systems came into being. And all of a sudden, we had a whole set of new insights into the process of apoptosis, which we lacked previously. That is the origin of many of these effectors, a link to these prokaryotic immune mechanisms, which had been taken as is by eukaryotes to establish many of the immune mechanisms seen in metazoa, again paralleling the emergence of the apoptosis system. So all that remained was to see how these came together in prokaryotes, and whether we could now say something about those domains which we found, the domains of death, so to say, which we discussed the last time, whether they came together with such systems in prokaryotes in some way, where we could bring them all together under a unified umbrella. So this was sort of the foundation which was the ground on which we developed these two new works, which sort of brought a closure to the prokaryotic side of the apoptosis story.
Right, right. I know this is not exactly sort of going with the science, or it's about the science, but rather, the sociology of the science, in the sense that, this paper that you last talked about, which was on the discovery of these nucleotide-centric systems, is perhaps one of the papers where, which is which has so many insights that are least cited. For example, I still remember the fact that here's where we make the prediction that TIRs are enzymatic, or the CRISPR polymerase would generate a signal, a nucleotide signal, or the TRPM channels and aspects of the slog domain and so on. And this perhaps was read, I don't know, but never cited afterwards for some time. And we had to literally, even though we had actually gone and presented this in many conferences and many places, I always have this gripe that this is a classic paper, but still did not. Of course, over time, I guess it just has reached the target audience. But we live for citations at times, and this is one where I feel could have been better cited by researchers and other researchers.
Oh, absolutely. I think I don't know if I should say we live for citations, because that would be a very dangerous thing if we made that.
Correct. No, I mean, in the sense that, it is a small sort of a small thing about our work being recognized by others, or at least seen by others. Forget recognition, at least being seen by others.
I'm pretty sure it was seen by all the relevant parties which failed to cite it.
Right.
That is pretty obvious from what ensued. And it is true that there's a lot of stuff in this paper, and perhaps there is a modern culture in science, unlike that from the past, where there is a low attention span. And so maybe one could say that not everyone got what was being said there. But as you said, one of the key things was indeed that prediction of the catalytic activity of the TIR domain, and the fact that it would be acting on that glycosidic linkage and clipping it off, which could happen in NAD to generate ADP ribose. That was the first time we made it, and I think a couple of years after that, it was vindicated by wet lab studies, which established that. And the same applies for its relative, the DR hide. And of course, earlier we knew that happened with the slog domain. We also had that collaborative work with Darwin's lab,
right,
where a signal generated from that in Mycobacterium tuberculosis was recognized. So there were all those studies, but it finally, as I mentioned the last time, the story of the TIR domain, we knew it was enzymatic for a very long time, but it was almost, I would say, 2 decades after I realized that it was enzymatic, we could put our finger on what exactly its catalytic activity was. Right. So in a sense, this work clarified a lot of what was going on in the realm of apoptosis. And since then, it has become very apparent that the TIR domains, which were seen as passive adapters, are enzymes, and both in plant and animal systems, that's being extensively explored now.
And so this lays the base for the 2 papers that came more recently from our group, trying to actually understand what these apoptotic domains were doing in prokaryotes. And I think, in a sense, it required such a, filling up the gaps from very many different studies like this to be able to actually set this up so that we understand the grammar of these. One of the things I think that you keep saying is that, it's trying to understand the grammar of these systems, which is a fascinating way of looking at biological systems in a sense.
Right. Because in the end, biological systems are linguistic systems. Mm-hmm. And much of the language of biology is very similar to our human languages. So the methods of analysis of biological systems should closely parallel that of human languages, which is something which our tradition has long paid attention to. And I would say was the first time ideas like homology ever came into being was in the study of the Sanskrit language uh, through a linguistic mechanism, and how parts of words can be considered homologous was discovered therein. So it's a very comparable activity which we do here.
This is like a good end point for this
Yeah,
discussion. That is laying the
yes,
groundwork for the current discoveries and
right,
understanding of
yes,
the apoptotic system. So, in a sense, we can stop right here for this, and then in the next one or two sittings, we will actually go through these new discoveries and in a more elaborate way now that we understand the syntax of these domains in these contexts.
right.
Okay.